Hello, everyone. Good afternoon, and welcome to Webinar Wednesday. Today's topic is in recognition of Lung Cancer Awareness Month, which is this November. And we are going to be talking about lung cancer research and patient advocacy. As a reminder, for those of you who are joining us on Zoom, please mute your microphone when you're not speaking so that we don't hear any background noise. This webinar is streaming to Facebook Live right now, and the video will later be uploaded to YouTube. For those of you who are on Zoom, please use the chat box to ask questions during the presentation. And for those of you who are joining us on Facebook Live, please use the comments section to ask any questions that you might have. My name is Olivia, and I work in the University of Chicago Comprehensive Cancer Center Office of Community Engagement and Cancer Health Equity. I'm joined today by my colleague, Erica Rodriguez. So for those of you on Zoom, if you have any questions, you can message Erica or myself, and we will be happy to help you. The Office of Community Engagement and Cancer Health Equity is a bridge between the University of Chicago Cancer Center and the research that goes on there and the larger communities that we serve both in Chicago and around Chicago. Our speakers today, we have Ivy Elkins, who is the co-founder of the patient advocacy group EGFR Resistors. She's a lung cancer patient and survivor. And Olivia Pura, who is a PhD candidate in biochemistry and molecular biophysics at the University of Chicago. So I'm going to stop sharing my screen. And I believe Olivia is going to be speaking first. So Olivia, you can go ahead and share your screen and get started.
Great, thank you so much for the lovely introduction. Hi everyone, I'm Olivia. Can you see the correct screen or is that my home screen?
It's your home screen.
Okay, great. That is not as interesting as what we're supposed to be seeing. There we go. How about that? All right, great. Hi everyone.
Yes.
How is that?
Yep, that looks good.
Okay, great, awesome. All right, so thank you so much for that introduction. As mentioned, I'm a PhD candidate in my second year now at the University of Chicago. And today I will be talking about just a little bit of the work I've been doing, kind of in the context of lung cancer and uncovering the role of copper specifically in lung cancer. So this work is with the Chuan Ha Lab, who he is affiliated with, I believe, the Cancer Center as well. So just a little bit of background. Heavy metals are, of course, necessary for life. So in our bodies, we actually have quantifiable levels of heavy metals like copper, nickel, arsenic, other things like that. And those metals are actually very important. We need them to live, we need them to survive, and they carry out a lot of really, really necessary, important physiological functions, like letting our cells communicate with each other, making sure our enzymes are functioning properly, and maintaining the structure of our proteins within our cells, within our tissues. Of course, because those metals are so important, the levels of those metals are actually very tightly regulated. There's a lot of mechanisms in place to make sure that those metals are maintained where they're supposed to be, and it's a pretty small window for most of those metals. As you would imagine, when those things become dysregulated, they actually can lead to a variety of diseases. And of course, we're going to be talking about cancer today. So there has actually been a lot of work done that has found that heavy metal dysregulation or dyshomeostasis actually can be a very important component of promoting cancer development. And this happens through a variety of pathways. So heavy metal imbalances can actually damage DNA. They can impair the enzyme function when those enzymes are responsible for detoxifying our bodies and maintaining the right levels of different substances in our cells. They can result in uncontrolled oxidation within our cells or within tissues, and they can disrupt the cell cycle. So it can cause cells to, divide more rapidly than we would typically want, and that of course leads to disease development. So those metals are really, really important when we're trying to understand cancer and why it happens. In particular, and this is the bulk of what we'll be talking about today, elevated copper levels in lung tissue have actually been linked to cancer development in lungs. So the idea is that when there is extra copper in the lung tissue, there is some sort of signal that is then relayed into the cell via these different cell signaling pathways. And ultimately, this results in some sort of change in the genome of the cell, and that actually kind of manifests itself as some sort of cellular response. This is really kind of the broad zoomed out version of the pathway that we're trying to understand and get a better, clearer picture of. Unfortunately, we actually have not yet found a single protein that is able to both sense copper and relay that information to the cell that results in that cellular response. So this is actually extremely important because if we know what protein is responsible for relaying that signal, that could actually present itself as a new target for cancer therapies, and of course give us just a more complete understanding of what's going on in the cell and in the tissue that's driving that disease. So our goal was to understand and try to find what protein was ultimately responsible for that. And we used a really, really interesting technology for this. So if we want to understand how lung cancer cells respond to copper levels, we can actually kind of do this through a bottom-up method. And there's this really fascinating sequencing method that was developed by someone else in my lab called Cas-seq. It's a lot of jargon, so please bear with me, but there will be an explanation of this as we continue forward. Cas-seq actually stands for carboxyl-assisted single-stranded DNA sequencing. And what that means is that we're trying to find single-stranded DNA in a cell and label it and then sequence it. And what this is actually useful for is that we're able to sequence active transcription in the genome. So single-stranded DNA corresponds to genes that are being transcribed. And when a gene is transcribed, that's ultimately representative of the fact that the gene is being expressed in response to some sort of stimulus, like copper in the case of lung cancer. So we can then know what genes are being expressed in response to copper, and kind of going back to that pathway, almost go backwards. We'll find what genes are activated here and relay them back through the pathways to try and understand which protein up here is ultimately responsible for sending that signal. So that's how we wanted to identify the copper sensing protein in these lung cancer cells. And as sort of a representative of what this technology is actually doing, this is what transcription kind of simplified looks like. There's a protein that is responsible for splitting open our double-stranded DNA and creating a single-stranded RNA that is ultimately transcribed into, or translated into some sort of protein. We're not really worried about the actual protein itself that is responsible for this, so we're kind of just gonna get rid of that and focus on what's happening in the DNA. Our Cas-seq probe, like I said, only binds to single-stranded DNA. We are then able to purify and fragment out that DNA. So we get rid of all this double-stranded stuff. And when we sequence it, that ultimately reveals what genes were expressed in response to copper stimulus. And we identified that EGR1 is more expressed in response to copper in these cells. And interestingly, EGR1 is actually a transcription factor that is known to regulate genes involved in oncogenesis and tumor progression. So what that means is EGR1 is a protein that binds double-stranded DNA and ultimately begins that transcription process. So this kind of made sense because in response to copper and those elevated copper levels, we have genes being expressed that lead to tumor progression. It actually has an interesting second function. It shares this sequence with another transcription factor, SP1, and SP1 is actually responsible for regulating the gene that eventually translates into a copper importer called CTR1. So again, all these things kind of were in line with what we expected to see. We found a really interesting relationship between EGR1 and this copper importer. But what was ultimately really, really exciting, the kind of so what of all this for us when we did this experiment was that, oh gosh, I'm jumping ahead. Sorry about that. We then did a second experiment just to confirm that this was actually happening in response to copper. This is just a graph to show, with higher concentrations of copper, we experience a very, very large change in EGR1. So it's actually 6 times as much EGR1 using this highest copper concentration. Sorry. So back to the big picture of the story. Why is EGR1 so exciting? It turns out that EGR1 is also induced by EGFR signaling. And this is where Ivy and I made a really interesting connection. We'll kind of discuss this later. EGFR has already been a pretty well-established driver in the non-small cell lung carcinoma. So this was really, really interesting because never had that protein been relayed to copper homeostasis. So this was actually a really, really interesting relationship that, hadn't really been investigated before. So we knew that EGFR was important in lung cancer, but we had no idea that there was a chance that it actually responds to copper in any sort of way. So this is kind of what we're currently investigating. We're trying to get a clearer picture of how the proteins and how the copper kind of interplay to drive the disease and what sorts of other players are involved in this. But we had previously just understood that EGFR was important in driving the cancer. We knew that that mutations in EGFR could lead to lung cancer, but we never knew that it had any sort of role in potentially, maintaining or unmaintaining copper levels in lung tissue. So what we currently believe and what we're trying to prove or maybe understand a little bit more clearly is we believe that EGFR is ultimately what binds copper. It activates those cell signaling pathways to increase EGR1 levels, and that ultimately drives the expression of genes that promote cancer development. Of course, that is just a hypothesis. We have a lot of work left to do to actually, be able to say that this is actually what's happening. But with a series of biochemical experiments, we'll be demonstrating that EGFR can bind copper. And ultimately, the important part of research is making sure that we're making an impact on, real life patients, real life, people that deal with this disease. And just proposing a mechanism and kind of leaving it at that isn't particularly helpful for anybody. So what we really want to look into is if we can find some sort of relationship between copper chelation or copper binders and chemotherapeutics that target EGFR to improve the treatment of the disease in these models where copper is upregulated and EGFR is potentially upregulated as well. So, there's a lot of really difficult disease types to deal with. And we hope that by combining our understanding of the copper mechanisms and the copper regulation that is involved in lung cancer development with what we already know about lung cancer, to come up with perhaps a new treatment model, a new treatment system that kind of takes advantages of both of the roles of EGFR to hopefully, help patients get the treatment that is going to work best for the type of disease and the mechanisms driving their disease. Does anyone have any questions for me? Because that's all I've got.
So I have a question, Olivia, and you sort of touched on this. Just kind of thinking about so can you just explain a little bit more? What are some of the real world applications? Once, let's say you're in the lab and you've proven what you set out to prove here in regards to copper and EGFR. So what does that mean with like real life, in terms of real life implications for people who are lung cancer patients?
Great question. So unfortunately, the road to that point in research is so long, especially because we're kind of starting from really the back end of kind of developing a better understanding of a pathway that drives a disease. But there has been some kind of speculation as to whether or not, removing copper from that system would help prevent or treat disease. And there's also already therapeutics that target EGFR. So what we really want to understand now is if we can combine those two treatments and understand how they work together to stop or treat cancer. That's really the goal, to kind of see if we've now shown that EGFR might have an additional role beyond what we already thought was the case with maintaining copper levels in the lung tissue. So we're really interested to see if by kind of removing copper from that tissue system and then also inhibiting EGFR, can we really kind of hit that protein in two different ways to really stop the disease progression. That's really the goal.
For people who are unfamiliar with EGFR, can you just explain a little bit about what EGFR is? I know that you touched on it a little bit in your presentation already.
Yeah, so on a cell surface, EGFR is a protein that actually crosses the cell surface. So it has a part of the protein that is sort of outside the cell and then a part of the protein that sits within the cell, which means that it's able to kind of relay messages across the inside and outside of a cell. So it's what's called a transmembrane protein. And it is a very common transmembrane protein present in a lot of different cells. And it essentially is able to interact with things that are outside of the cell, different ligands outside of the cell, molecules, things like that. And then when it kind of interacts with those things that are outside of the cell, it's able to then relay that message into the cell and cause some sort of reaction based on what is happening outside of the cell environment.
There's a question here in the chat. It says, how are you all testing or screening for copper that might cause factors? Are there any signs that one can look out for?
I don't think I understand what that question is asking, in the sense that are we trying to develop a screening method for copper levels in patients? Is that kind of what the question's getting at?
She's asking, is there already a screening? Is there screening for people, or like, are there physical symptoms of someone that might have this?
Not that I'm aware of, to be honest, but I think one of the downsides, unfortunately, of being so buried in the lab is I don't only get to see the clinical side of lung cancer. So I'm honestly not sure. Maybe Ivy could be a little bit more insightful on that question. But on my bench side end, I have no idea, to be entirely honest.
Yeah, and I do know that there are, screenings for lung cancer in general for people who meet certain criteria. I don't know that it is specific to measuring the amounts of copper that people have and where it's binded to, or, I don't know that it gets that specific.
Let me jump in for a second. It definitely doesn't get that specific at this point, but that's, the power of the research that Olivia and, others might be doing. There might be information that comes out of this research that might improve our screening processes that we have right now in the future. And this is Ivy, by the way.
Exactly. Great point, Ivy.
Awesome.
Olivia, I have one more question for you. Just can you give us a little bit of your background and what brought you to this point in, you're a PhD candidate, you made this really big commitment to work in a lab 24/7 and really, focus on some of these really, molecular issues when it comes to cancer. What was sort of your inspiration for choosing this line of work? Did you have a personal connection to cancer? Just explaining your background a little bit.
Oh my gosh, I don't even know how to answer this question succinctly. I guess I'll kind of start like way back when. I actually lost an aunt to oh gosh, what was her cancer type? It was an eye cancer. It was a carcinoma in her eye that ended up spreading, and it was a really difficult experience to have as, I think, a 12-year-old at the time. And my whole family lives in Poland. That's where my parents are from. So it was just really hard to lose someone and to see how difficult it was to deal with a disease, not just because it was cancer, but then, having someone so far away in distance and not being able to kind of be at her side as much as we would have liked to be as this was happening, really left an impact on me as a child. And I actually started college at Northwestern as a pre-med. I thought I would go to medical school, kind of figured out through the process that I was a lot more interested in kind of the behind the scenes of disease, understanding why disease happens, how we can actually use our understanding of like really nitty gritty science to try and improve how we're able to treat disease and how we're able to develop different screening methods or treatment methods. So I knew I was interested in the medical space and kind of the medical field generally, but it wasn't until college that I got to be in a lab and talk to people about science in kind of like, again, that nitty gritty sense. And really kind of work at it from the back end so that hopefully, down the line, the work that we do at the bench is able to help people, in the clinic. And that's really kind of the goal.
That's a great backstory and a great explanation. Do we have any other questions for Olivia and her research before we move on to hearing about Ivy's story? Okay, thank you so much,
Great.
Olivia. And if you could just kind of stay on, because I'm sure we'll have some questions for you and Ivy after she does her presentation. So Ivy, you can go ahead and share your screen when you're ready.
Okay. Let me. bear with me 1 second while I
Sure thing.
get this up. Okay, there we go. Can everyone
good.
see my slides now? Okay, great. As Aaliyah mentioned before, my name is Ivy Elkins, and I am a lung cancer patient and advocate. And I am actually living with the EGFR positive lung cancer that Olivia is studying the connection between with respect to copper. And I have been living well with stage 4 EGFR positive lung cancer for almost 8 years now. The only reason why I'm alive today is because of research. Research like Olivia is doing is a lifeline for patients like myself because we depend on new discoveries to lead to treatment options that allow us to continue to thrive. I'm going to share my story with you today, both as a patient and as a research advocate. There we go. So I live in Evanston, Illinois, just north of downtown Chicago. I've been married for 25 years and have 2 sons, currently 18 and 21 years old. When I was diagnosed with stage 4 EGFR positive non-small cell lung cancer in December 2013, my boys were 10 years old and 13 years old. The first picture up on the screen there was taken just about 1 month prior to my diagnosis. In the middle of 2013, I started having some pain on the left side of my neck and difficulty straightening my elbow. I didn't think much of it at the time, but as it didn't go away, I went to my primary care physician and he diagnosed me as just having an overuse injury, probably from too much use of my computer. I went to physical therapy, and over 6 months, I visited a couple of different physical therapists, 2 different orthopedists, a rheumatologist, and tried numerous exercises and cortisone shots for my elbow, which just wasn't responding to what they considered to be normal treatment and kept getting worse. Finally, I had an MRI of my elbow, and the MRI showed that there was a mass on the bone, which had partially eaten away at the joint. I then visited an orthopedic oncologist who performed a biopsy and found that the mass was malignant, a form of cancer known as adenocarcinoma, which Olivia mentioned before, which doesn't originate in the bone. So I had a PET scan, and it turned out that the origin of the cancer was a tumor in my right lung that had spread to my bones. That's why I had the trouble, of course, with my elbow, but also my neck. And I had some metastasis in my ribs and my pelvis. And a subsequent brain MRI showed that I had 8 small metastases in my brain as well. I cannot tell you how shocked I was to find out that I had lung cancer at age 47. It was not on my radar screen, and I didn't have any of the symptoms that I associated at the time with lung cancer, like coughing or wheezing or shortness of breath, nothing. I really didn't know then what I know now, which is that anyone with lungs can get lung cancer. I also, and this was a big deal, I didn't know that not all lung cancer is the same and it's not treated the same. Fortunately, I found a fantastic thoracic oncologist and found out that my lung cancer was driven by a specific mutation in the EGFR gene. And this is where my path changed dramatically. As a result of that, I was able to be treated with an oral targeted therapy medication. It's a pill, I take once a day at night. And I have been treated with two different targeted therapy medications for the past 8 years. And they specifically target my EGFR mutation. My first targeted therapy medication worked for almost 3 years. That's the downside of these medications, is that eventually, usually, resistance develops to the medications. And after 3 years, I had a little progression in my original lung tumor. And thanks to research, there was a new targeted therapy for me in 2016 that had just been FDA approved. I had a fantastic response to these medications. My brain lesions disappeared, bone healed over and grew back in my neck, my elbow, and other areas, and the main mass in my lung shrank significantly. In early 2020, just a couple of years ago, I had a PET scan that showed the only area of identifiable cancer I still had left in my body was the original primary tumor in my upper right lung. So I actually had a lobectomy at that point to remove my upper right lobe, and I've been considered to have no evidence of disease for the past 2 years. None of that would have been possible or would have happened if I had not found out that I had an EGFR mutation. Research matters so much. When I was first diagnosed, as I mentioned, the medication I take daily today didn't exist. I never thought I would become eligible for surgery, let alone become NED, have no evidence of disease. And those 2 little boys in that first picture up on your screen, they're now college students, and I've been able to live well with lung cancer while raising them with my husband. Oops, I went the wrong way. There we go. Sorry about that. So I'd like to go back a little bit to something I touched on while telling my story. I mentioned that when I was diagnosed, I thought all lung cancer was treated the same. And, obviously this is not the case, and much has been learned about the different molecular types of lung cancer over the years. Adenocarcinoma, the type of lung cancer I have, makes up about 40% of all lung cancer diagnoses, and it's a type of non-small cell lung cancer that is biomarker driven. The pie chart on the screen is a bit difficult to read, but I put it up to show a point. And it's important because it shows all the different biomarkers that have been discovered to date. EGFR, the big blue wedge on the bottom of the pie, represents about 30% of adenocarcinoma diagnoses. And different biomarkers can mean completely different treatments, precision medicine treatments that directly target the changes that cause cancer. I was incredibly fortunate that when I was diagnosed, I was at a large academic medical center in Chicago and was automatically tested for the presence of biomarkers to direct my treatment. This really is not the case everywhere. And not everyone automatically has access to this potentially life-saving and extending biomarker testing. This creates incredibly unfair disparities in healthcare. In order to help combat this inequity, Illinois passed Bill 1779 this past August, which expands coverage for biomarker testing. Starting in 2022, biomarker testing will now be covered under Medicaid, as well as state-regulated insurance plans. To date, this type of biomarker testing mandate has only been approved in a few states besides Illinois. I believe just California and Louisiana have passed somewhat similar bills. The hope is that more states will follow, or there'll be some action at the federal level so that everyone can get the biomarker testing that might improve their specific treatment for lung cancer. Sorry, I'm having trouble with oh, there we go. So biomarker testing and the therapies that are targeted to these biomarkers have really allowed people with lung cancer to live longer and better in the past 5 or 10 years or so. And as a result, they've been able to take a larger role in advocating for themselves and others. So in the past several years, advocates like myself have formed patient groups for many of the biomarker-driven cancers on the pie chart you saw just a couple of slides back. These groups are usually patient-run and determine their own agendas, focusing on what they feel are unmet needs in their subset of lung cancer. One of these groups is the EGFR Resistors, which I co-founded with 6 other patients and 1 caregiver in the middle of 2017. we decided that we wanted to build a community of patients and caregivers to share knowledge, provide support, and work to accelerate research with the goal of improving outcomes for EGFR positive lung cancer. We created a closed Facebook group for patients and caregivers only and communicate with the broader EGFR positive community through our website, Twitter, and Instagram. We have a monthly newsletter that anyone can sign up for on our website to receive. So I encourage all of you to do so if you're interested in learning more about our work. Please also feel free to follow the EGFR Resistors on social media. Our initial group of 7 founders has grown to over 3,100 members from more than 80 different countries by now. Unfortunately, many of our members have experienced resistance to the current standard of care treatment, including several of our original founders. This is why increased research is so important. 4 of our founders, you see pictures up on the screen, Terry, Anita, Alan, and Ildi, have passed away in just the past 2 years. This underscores our critical need for research and new treatment options, and we continue to advocate with increased urgency in their honor. The EGFR resistors recognize that many challenges exist that our members often need to overcome in order to get the best possible treatment. In order to help our patients overcome these challenges, we do much more to help in addition to providing support. We increase awareness and education for our members in a number of ways, including providing information and firsthand experiences with treatment and trials, giving tips for communicating with healthcare teams, sharing information about symptom and side effect management, and just help with asking the right questions. All of this assistance builds confidence for patients to take charge of their care and advocate for themselves. Many end up becoming lung cancer advocates and advocating for others as well. We also help improve access to effective diagnosis and treatment for our members by sharing information about important issues such as getting second opinions, having biomarker testing, and having repeat biopsies upon progression. We suggest resources to assist with cost and insurance coverage issues and share information about clinical trials. In this fast-paced world of new developments in lung cancer, patients and caregivers, especially depending on where they live, often need to be the ones to bring information about testing and treatment to their doctors. Last, but certainly not least, we've created many collaborations in order to fund and accelerate research, patient-directed research, and we've formed these collaborations with researchers, large nonprofit organizations in the lung cancer community, such as Longevity and Lung Cancer Research Foundation, various pharmaceutical companies, and a continuing medical education provider called CEC Oncology. In the interest of time, I'm just going to discuss one of our collaborations in detail today because it directly connects to the important work done by researchers like Olivia. Through our partnership with CEC Oncology, the medical education provider I mentioned, we have held annual EGFR Resistors research summits in 2019 and 2020, and we have our 3rd one coming up this coming weekend. Young investigators are invited to apply to the summit if they are doing research in EGFR positive lung cancer, and if accepted, they get a chance to present their work in front of our mentor judges, who are well-known leaders in thoracic oncology, often specializing in EGFR positive lung cancer. We have winners for best presentation, and participants get small monetary grants to recognize their work, which hopefully will lead to future larger grants from others. But the true reason and the true benefit of the summits is the development of collaborative relationships between the young investigators and mentor judges, and we want to promote these. We divide the young investigators into squads to work in small groups with EGFR experts, and these experts mentor them in things like grant writing and publishing and clinical trial development and other, important areas. From the patient advocate perspective, we want to encourage these young investigators to continue research work in lung cancer, especially EGFR lung cancer, and collaborate early in their careers. So that's why we do this. And, we know that encouraging their research is in everyone's best interest. And as patients, it's our lifeline, like I mentioned before. So in summary, I'd just like to emphasize how important it is for all of the parties on this screen to work together in order to move forward. Oncology really is a team sport, and we need the involvement of all these groups in order to make the urgent strides in research and development that patients need and deserve. Programs such as the CORE program that Olivia and I are part of are so important because they bring together researchers and community members, and they create spaces for us to speak with each other and learn more about the challenges and benefits we each face. So thank you for listening to me today, and let me know if you have any questions.
That was great. Thank you so much, Ivy, for sharing all of this. And we have a note in the chat here. Ivy, thank you for sharing your story, and prayers for continued progress in your care. And I think everyone on here would certainly second that.
Thank you.
Ivy, so I'm really just so impressed with what EGFR Resistors has been able to do in a pretty short amount of time. How did you, when thinking about forming this group, how did you find other people? It seems, like such a niche sort of thing. How did you find other people that you could work with this group that had EGFR positive cancer?
One of the first things that happened when I was diagnosed was I was introduced to another patient in the local area who also had EGFR positive lung cancer and had been involved in advocacy for a while herself. And between the two of us, she encouraged me to attend some various events such as Longevity, which is one of the large lung cancer advocacy groups, has an annual Hope Summit. So I went to that and I met other patients. And just through getting involved in the lung cancer community and, participating in some conferences and things like that, I came to know other people who were living with EGFR positive lung cancer. So that's really how the 7 of us initially found each other. We were each connected. It was almost like a game of telephone. We were each connected to 1 or 2 of the others, and we talked and felt that there was a need for a group. And as the original founders, we really had a good mix of different background and abilities between management and marketing and medical and, social media experience. So it was a really great combination of people to get started. And the power of social media now is huge. So that's how we've managed to connect with people all over the country and all over the world. And it just kind of took off from there.
Great. And can you tell me a little bit about what do you, meeting with, as Olivia mentioned, she's, I'm in the lab, I don't interact with patients, and patients don't interact with basic scientists most of the time. And so what do you wish that researchers like Olivia would know about people who are lung cancer patients or survivors or living with lung cancer?
It is so wonderful to meet researchers like Olivia because it just humanizes the whole process on both sides. So, I think, I would want researchers like Olivia to understand that, we're, just people. We could be your friends, sisters, brothers, parents, whatever, we're real people and we're all individual and we all have individual goals and interests, but that also goes both ways. From the patient advocate perspective, we think of research, with a capital R because we don't meet researchers that often, especially basic science researchers who are, in their labs most of the time. And, through connecting in a way like this, we get to understand them as people, and we understand what they're interested in and what makes them tick. And it also helps patients like me understand sometimes why research takes a while, because you'll read something in the paper about some huge discovery in a mouse model or in a cell line and think, okay, well, when am I getting this medication? But as a result of collaborations like this, it's easier to understand that these things do take a while. And why? Because it's a very, very intensive process. So, I think from the point of view of patients and patient advocates, I'd like researchers to know that we can understand these things and keep explaining them to us, just use less complicated language, but we're interested, we want to learn. A lot of us want to learn about discoveries, and we are so excited to hear and get to know you and hear about what you're working on, because like I said before, it really is our lifeline.
Yeah, absolutely. I think, a lot of researchers maybe don't realize how engaged the patient population can be around the work that they're doing. And so, Olivia, same sort of question for you. When you met Ivy, how did that sort of change your perspective on what you do in the lab, and how will you take some of that and move forward with your work?
Well, I think one of the biggest qualms I've had, I guess, with being involved in translational research is that we don't have opportunities given to us to meet with patients and to meet with people that actually benefit from research. And, we kind of have to seek out those opportunities ourselves. So, obviously I'm very fortunate that through U Chicago and through the Comprehensive Cancer Center, I was able to meet Ivy and to have this incredible conversation about how, the work I'm doing, kind of relates to her experience and her cancer survival story. And it really just puts kind of that extra fire under our butt sometimes to remember that we're not doing this just to publish a paper or to get into a cool journal or to, get my job in a couple of years. And oftentimes that's kind of the mindset, unfortunately, in academia. You do research because it gives you something to put down on a piece of paper. And obviously not everyone is in that mindset, but I think kind of the overarching idea is, publish or perish, because there's always more to do and always more to find. And I really kind of struggle with that mindset. Like I said, I originally, as I started my, college degree, I thought I wanted to go into the medical field as a physician. And I think the mindset in physicians and in medicine kind of more specifically is a lot more patient-centered and more people-centered. Whereas academia and being a PhD student, it's certainly not the case. So, meeting with Ivy and meeting other, survivors and other advocates serves as a really necessary reminder to kind of hold on to the real reason why I'm here, and not the reason that's being kind of prescribed by a lot of other people that do this. So it's really impacted me in an incredible way, and just reminded me why it's so important to do what I'm doing, even if my paper doesn't get into one of the top 3 journals, or even if, I'm not cited 1,000 times. That's really not the point. The point is to do something that will ultimately save someone's life one day. And that's so much more meaningful to me.
That's an excellent perspective and point to bring up. Ivy, what would you recommend? So you're, lung cancer specific and EGFR specific, but what would you recommend to anyone who's been diagnosed with lung cancer and is like, or I'm sorry, been diagnosed with cancer in general, any kind of cancer, and is like, I want to get involved in patient advocacy and I want to get involved in research advocacy. Where can they start, and what do you recommend that they do to sort of get going? Because when you're not involved in the cancer space, before you're diagnosed, usually there is a pretty big gap in the knowledge and what you know.
Yeah, I started really by reading articles. First, my initial, foray into advocacy wasn't even that I was thinking about being an advocate. I just wanted to learn more about my type of cancer and how it formed and what potential options were on the horizon. And, just so you know, I have no background whatsoever in science. I have an English and finance, degree, so nothing related to science. But as, I started reading more, I, realized that, I really, really had a strong interest in things. So I signed up a lot of conferences, have rates or even free for patient advocates. Luckily, ASCO, the American Society for Clinical Oncology, takes place here in Chicago. So I went to an ASCO meeting and kind of tried to listen to things and understand them and introduce myself to people. And I also got involved, like I mentioned before, with Longevity as a large, nonprofit patient advocacy organization, and made connections through there and learned more through that involvement. They actually, they knew that I was interested in patient advocacy, and they tried to find opportunities for me. Initially, more telling my story, but then as I learned more about research, providing my patient perspective on research. So I guess anyone who is interested, I would recommend really to read, to attend things, and to look for organizations related to your specific type of cancer to find some opportunities where you might be able to really add value as a patient, because we have a perspective as patients that's really needed in research. Research is great, but we want it, researchers want their research to meet, what we need. So, it's important for us to be in there and sharing, what we need and what might be drawbacks or what, might not work well about certain things. So I encourage anyone who's interested in advocacy to get involved. And you can be involved in a lot of different ways, not just necessarily getting involved in research. There's lots of ways to be an advocate depending on what you're interested in.
Great point. And so Ivy actually spoke on a webinar earlier in the year. I think it was May, maybe, it was for excuse me, Cancer Research Month. And I've kind of totally had forgotten that we put together, based on what you and our other presenters had talked about, we put together a cancer research advocacy toolkit. And I just dropped the link in the chat box. And when we send a follow-up email to our Zoom participants, we'll include it there as well. And I'll drop the link on our Facebook page as well. But this was a toolkit that kind of put together some of the resources that Ivy and another patient advocate, Kimberly Richardson, had talked about on that webinar. So if anyone is interested in getting involved in patient advocacy and specifically research advocacy, as Ivy said and Olivia said, it's just so important for those two parties who are not necessarily, in constant interaction with each other, right? Like Olivia said, a patient is always kind of interacting with their clinician, but not with the researcher who's doing a lot of that bench and basic science research that's going to develop the treatments that can keep someone like Ivy alive and with no evidence of disease all these years later. So making sure that those two parties stay connected is really important. In the interest of time, so if anyone else has any more questions for Ivy or Olivia, please use the chat box, unmute yourself. For those on Facebook, again, use that comments section. And I just want to kind of ask Olivia and Ivy for each of your main takeaways regarding patient researcher relationships and advocacy.
Olivia, do you want to go first?
Sure. Gosh, I think my main takeaway, maybe this sounds so simple, it's just so necessary. It's not something that's patient researcher interaction and collaboration isn't considered standard by any stretch of the imagination, especially kind of more on the basic science side of things. So I think my biggest takeaway is simply that we need to work, we meaning, us, the researchers, need to work to create these relationships and to maintain these relationships throughout our journey in science. And I think it starts, with people kind of at my level, at the PhD level, but it also starts with people at the upper level and kind of the people that dictate what sort of science goes on and how that science goes on. So it's really just making these things happen is so important.
I completely agree. And it just demystifies the whole process from the patient point of view. When you're, diagnosed, you're thrown into this whole new world with new terminology and, a lot of it you don't initially understand. And personal connections at that point are so critically important, both at the beginning and throughout the whole process. So just making that connection between researchers and patient advocates are so important on a human basis, and also in order to move things forward, because we provide the kind of urgency, that is needed, because we're real people experiencing resistance and, side effects. And, it's just important for us to keep our communication two ways.
Absolutely. Great points by both of you. Thank you so much for sharing that. I did want to share one quick infographic, and Ivy, this was one that we found from Longevity for the webinar we had the other night, but I think, okay. Okay. Hopefully you can see my screen okay. So sort of the driving factor behind a lot of this too is that lung cancer accounts for 12% of all new cancer diagnoses, but 22% of the cancer deaths. And we see in this pie chart in the middle here that lung cancer takes more lives annually than the next three most common cancers combined, which are breast, prostate, and colorectal. And so getting more research on, more people like Olivia in the lab who are connecting with patients who understand the urgency behind the research that's going on is just so critical. And the bottom half of this infographic I really like, because it just shows that, so first of all, only 6% of federal research dollars are spent on cancer research are spent on specifically lung cancer. And so this little chart on the left here shows the difference between the number of cancer deaths, which is represented by the little blue person, versus the number, the dollars spent per life lost, which is represented by the dollar sign here. And we see how great the disparity is in lung cancer, where we're losing a lot more patients than money being spent on life-saving cancer research. So this is really important to be thinking about during, especially Lung Cancer Awareness Month, but all the time, how much research is needed into this disease. And we are so grateful to Ivy for sharing her story. It takes a lot of bravery to share your cancer story, and we're just so grateful for you and all the advocacy work that you do. And we're so grateful for people like Olivia who are in the lab and trying to make these connections and these come up with new treatments that can work on our lung cancer patients. So thank you both for joining us today and for speaking today. We're so grateful that you're here and sharing your story. Thanks again.
Thank you.
Okay.
Yeah, thank you for having us.